Thanks for joining us for another episode of the podcast. I am so excited. We have with us here at the University of North Florida, and I'm going to make sure I'm saying everybody's name correctly, researchers Dr. BRYAN KNUCKLEY.
Yep.
Say BRYAN KNUCKLEY. We've also got Dr. Corey Causey and Dr. FATIMA REHMAN. Yeah. Thank you. And they have been researching ways to turn cancer off. This is what is so exciting to me about your research. First of all, tell me, what is it that you have found? What is this research? Is there a way to explain it to me where there aren't so many moving parts?
So inside our body, there are rules about when certain proteins are going to be made, when certain functions are going to be performed, and when they need to be shut down. So in this case, the PRMT are those regulators that are going to dictate when certain things are going to happen and when they will be shut down. And so when they go dysregulate, when they're dysregulated, when they're not working according to how they're supposed to be, then you get cells that don't listen. And that's when you get diseases like cancer.
So you are essentially getting a hold of the police officers in our bodies that are directing the traffic and directing things about how they're supposed to happen in our bodies. And there are some cells that are not listening to those orders, and those are the cells that turn into diseases.
That is partly true, yes. Okay. In this case, the police officer is not policing correctly. So the PRMTs that are supposed to mark certain parts of the DNA at certain times and have it function in a nice, well-oiled machine, do not do it appropriately. And if they are overexpressed, they are going to mark these areas incorrectly, and they are going to either overexpress proteins that cause extra growth, which would be examples of oncogenes that can cause cancer, or they will shut down even proteins and their expression that can suppress tumors, called tumor suppressors, because they are also regulators of cell cycle or other mechanisms like that.
So these rule makers,
yes,
when they go on the fritz and they're telling cells to do wrong things, you are taking those rule makers and shutting them off.
That's right.
Yeah, I think that's fair. Yeah.
And so how does that stop cancer? How have you found that that affects that bad part that is turning into the cancer? How do you take that rule maker and make it do what you want it to do?
So what has been found is that in a lot of cancers, these particular class of enzymes, the PRMT1, are overexpressed, which means that they are made more than normal. And they are, because they're made more than normal, there are more of them around, and they are going to mark the DNA in a way that it should not be marked. So you are going to get dysregulated growth and dysregulated differentiation. A lot of cancer is because of that. That is a key hallmark of cancer, is the dysregulated growth. And so when we shut it down or inhibit it, its activity, it is going to bring the body back in balance, that cell back in balance, and it is going to stop that dysregulation from happening, that growth from happening.
How did you find this? Was this something you stumbled on, or did you have a concerted effort like, I am going to study how to stop cancer, how to get rid of cancer?
I think we probably all have some goal of that, but different angles towards it, right? Because cancer is so complicated and there's no one area to focus. Mm-hmm. So I think like in terms of my research when I came to UNF was studying this one group of proteins and understanding how they worked. There wasn't a lot of research in terms of what they do in your body, how they work, and how sometimes they can be overexpressed or become too active in your body. But we knew that they had some role in cancers. And so our goal was always to just study and understand more about these proteins. And kind of one thing led to another where we always have an interest in trying to develop something that's going to turn that off. And over that course of time, I guess, we stumbled into something that kind of helped us say, hey, wait, this actually might turn off cancer. And I think that's where this collaboration really grew, is like, okay, we can actually test this and see if it's going to work.
So, and I know you were the lead on this, BRYAN, so what was that moment like when you were like, holy crap, look what we have here? Tell me about that.
It's actually an odd story. So a lot of times in research, things don't go the way that you expect. And it's kind of those moments where something failed, where something else comes out of that. So my lab was studying how these proteins work. And so we typically study these peptides, which are just a small piece of the protein. We were studying to understand how this protein works using peptides. And we turned to what we call these peptoids. So again, if you think of the Lego bricks coming together to make a protein, and we're just making a short fragment called a peptide, we now use generic Lego bricks. So instead of using the traditional, brand name Lego bricks, we made one that's kind of just a little off-brand, the generic Lego brick, and we put those together. And so we thought it would work just like the normal, brand name Lego. When we did that, we found that it didn't work the same. And so, in all honesty, my student who was working on it was disappointed. I was disappointed. I was like, well, that doesn't make sense. It should be working. Like, I don't understand why this wouldn't work just like the regular one. And that, I still remember that night going home and just disappointed. I was like, well, that was a waste of time, because this was months of work that it was like, okay, we're going to have a really cool result here. And when it didn't, it was just disappointed. And it may have been like the next day, a day after, when I started thinking, like, it doesn't make sense that it wouldn't work. And I remember, I think, going to Corey and saying, it's odd this didn't work. I was like, what if it's inhibiting? What if it's actually turning off this protein and we just don't recognize that? And so at that moment we tried it, and my student tried it. And the first time they came to me with the, hey, here's the result, it was like, oh my God, that is working. And so that was that moment where it was like, we might actually have something that might actually do what we would really hope to work, but we never expected that at that moment.
So you were just studying to find out more about the protein itself.
Yep.
And then in that fail, which wasn't, you realized that you had actually found a way to control how the bad was being created in the body and shut that process off.
Maybe at that moment I didn't know that much. I knew that I could affect this protein. And then it was kind of a couple iterations after that, or kind of working through that then it was like, okay, we need to test this, but I don't have necessarily that expertise or those types of supplies there. And so that's where we started working with Fatima to figure out, like, how could we test this?
And so is that your specialty? Is
Yes.
the testing part?
Exactly. So, that's what I do. My PhD was in cancer biology, and for the last 25 years, that's what I've been doing, is looking at different compounds and seeing if they cause cancer or if they can actually be used as targeted therapies.
So when you heard about that,
I was excited. I was like, yeah, sure, come on over, give us some compound, we'll test it. And so, my students and I, we got a little bit of his compound, and we tested it first just on one cancer cell line, a breast cancer cell line, because, we were like, okay, this is growing in the lab currently. Let's go ahead and start with it, test it out, see if it's even worth it. And the first time we did it, yeah, it seemed to have some effect, and it seemed to reduce the viability, and we were excited. So we were like, okay, let's try out some extra ones.
when you tried it that first time, did it stop it, or did it, what did it do?
So what it did was, the first couple of days, we didn't really see much difference. But then after that, we came in the third day, and we saw that there was massive death. So there were a lot less cells in the ones that were, cancer cells that were treated with this compound. And we were like, oh, cool. There's definitely some effect going on. And so we're like, okay, let's see if we can replicate it, and also add on some other cell lines, other cancers, and see if it can be replicated in those as well. And lo and behold, it worked on all of them. And so we're like, oh, cool. With some more efficiently than others, but definitely worked on all of them.
It showed at least some of the cancer was
Yeah. Definitely. It was either killing them or stalling them, but it was having an effect. And we could actively see the cells exploding through what we call programmed cell death, or apoptosis. And we could see cells that were showing indication of another process called autophagy, where the cell thinks it's getting starved and kind of eats itself up. And so we're like, ooh, cool, that's fun. So my student loves watching those cells that look like they're eating themselves up, because they're, cool. So we were like, ooh, that's fun. So, that was all well and good, but we were still worried that what if it does the same to normal cells? So then we worked together, and we got some normal cells. I had a few with me, but we wanted to get some really primary cells that were straight from the body. We got a couple of cell lines, and we examined them in there. and it didn't do the same to them. It was very targeted towards the cancer cells and not in the normal cells. So with the normal cells, they seemed to be just fine, and maybe slowed a little growth, but didn't kill them. They were fine. They were happy.
So it recognized the bad.
It definitely recognized something that was going on differently in those cancer cells. Maybe just because there was so much overexpression of this protein. Maybe there's something else that we also need to study, which we are in the process of trying to decipher. But it recognizes that there's something different here, there's more expression here, and when it inhibits it, you see that dramatic effect.
Wow. And then how did you get involved? You're the quiet one over here.
So my work, Brian and I have actually worked together for many years, and I'm a synthetic chemist, and so a lot of the molecule design and synthesis is my part of it, and then thinking about how these things work together, like on a molecular level, and then it sort of works its way up to bigger molecules and then actual living cells. And so I think the collaboration has been very effective. We see things from different perspectives.
Exactly.
I'm very micro, and then it moves up to actual living things. And so I think we all have a perspective to contribute that helps us problem solve. When fails turn out to be successes. It's very easy to throw things in the trash bin when it seems like the experiment hasn't worked, but until you take the time or have a different set of eyes on it, think, well, maybe we should approach it this way. And what Brian was explaining wasn't that the experiment failed, it's that we weren't looking for the right thing or didn't think to look for something else. And so that could have easily been put away and never seen again. And so when you have more heads approaching the problem, it's easier to find these things that might have otherwise been missed.
So what was your reaction when you heard that this was something happening here?
Well, I think it's one of these things where, you asked initially what the goal was. And I think, you've asked most people in our field to cure cancer, right? That's what everybody wants to cure cancer. But I don't think many of us think that, okay, it's a long drawn out process. And so the incremental steps it takes along the way. And so I think, that discovery going from, well, this seemed like a fail to that was actually a big leap forward. We imagined that one day we would get there, but that's a big leap. And then to go from there to actually seeing an effect in the cells was, I don't want to say I'm pessimistic. I'm cautiously optimistic, right? Yes. And so
exactly.
to see it work so effectively was it's pretty cool, right? And this is the idea of, well, this is really potentially effective, and these are real cells that are dying in real time, and we can experience that. And so it's one of these things, again, with being cautiously optimistic, but we might really be on to something.
So realistically speaking, I think, oh my gosh, they're killing cancer. We can get this in bodies now, but this is going to take a long time. So tell me like a time frame. I know you don't have a crystal ball, but are we talking like months? Are we talking like decades? What are you thinking?
So it depends upon the compounds and how quickly we are able to develop it and how we can get it into a clinical trial ready state. But from bench to bedside can take anywhere from 5 years to decades. And there are compounds that took 30 years to get there, but with modern advances and the way science works now, that time frame has been shortened. Each year it gets further shortened. So who knows? It depends on how quickly we are able to test it out further, actually understand the mechanism of action behind it, and then develop it into a molecule that can be delivered into a human being to test it out.
Do you have a goal that you would like to try to get to a certain point by?
I think that there's really two goals at the moment, I guess. One is this current compound that we have is really good, but we probably want to make it even better. And so we can do different chemical modifications to try to improve both its potency to these cancer cells, but also making sure that we're really selective, because the more selective you are to a specific type of cancer or a specific protein, making sure that you don't hit anything else in the body is outstanding, right? Because then you lose all those side effects. So that's one goal. The other goal is to kind of move it forward at the same time, right? So we've tested it in these isolated, cell lines. But really kind of the next step is moving it into more of a living system to understand how well it works. So that could be something like mice. I've had some discussions about trying it in zebrafish. You can have these different models of zebrafish or mice that have these different cancers, and then put these drugs into it and try to see if it actually affects them.
Okay.
So.
All right. I was reading why these 3 specific cancers. I read that you were able to see that this had an effect on breast cancer, type of breast cancer, lung cancer, and then the last one was colon cancer. Why those 3? Is it only these 3?
So there's a large amount of literature available for these 3 showing that they are definitely formed as well as progressed because of PRMT1 overexpression. And these were the ones that we had readily available in our lab as well. However, it is not limited to these cancers. It would definitely work on any cancer that has PRMT1 overexpression at an activity level or just at the gene level. And it would work similarly on all of them. So it's not limited to that. And that would include like esophageal cancer, thyroid, lymphomas, myelomas, several other types.
So am I understanding this correctly? As long as the cancer is caused in the same way that this can attack.
And not only those, there are actually some neurodegenerative disorders that have PRMT1 overexpression involved in there as well.
So not just cancer.
Not just cancer, as well as some cardiovascular disease components. So it can work in more ways than one. It is not exclusive to just cancer alone.
So what others would you?
So again, our focus has been on cancer, and that's what we would limit to. But again, it is targeting a molecule, an enzyme. So any disease or condition that is because of that overexpression would be amenable to testing.
What others are overexpressed in that way? I don't know.
Well, PRMT1 overexpression, like I said, has been shown in some neurodegenerative diseases as well. So like Alzheimer's, for example. And so you could test it out in those to see if it maybe increases the time to progression, because it's not going to completely stop it, but it can maybe slow the disease process in that case. People that have higher risk of heart disease or stroke, in some cases, if they are, nowadays a lot of our medicine is going toward personalized medicine. If they show that their, clinical etiology is because of PRMT1 overexpression, that could be something that could be used for that as well.
And that just made my heart just the idea that there's something that could work for something like Alzheimer's, and I mean that's big deal to me. My mom has dementia, so I hear any kind of study that's going on like that, I'm like, oh my gosh, this is so great. So sorry. It's just good to hear. So I apologize. You got a patent for this. What does that mean? And I looked up how to get a patent. That takes a really long time. You gotta convince people that you actually have something here, and they may not think you do, and then you go through this panel, and it's a long, drawn out process. So, what's involved in a nutshell, and why is it a big deal to have gotten one?
So we started the process in 2021 to actually get the patent. So you're right, it is a very long process. It takes, that initial application really is just explaining what you have and why it's so special, and that there's no one else out there that has anything close to it. That takes a lot of time to just develop that application. Then it goes through rounds of reviews, and people review to look at anywhere else in the literature someone has something similar, because you can't, have a patent on top of another patent. And so I think it takes a while.
What does it do for you? You have a patent, what does that mean?
Basically, it protects the intellectual property. So it's an idea, and it says, with this patent, no one else could capitalize on it while it's under patent protection.
But it also provides us with, in a way, it opens doors for us. It opens opportunities for us to foster collaborations that will allow us to develop it to that next stage, to be in the market. It will shorten that time from bench to bedside by a great deal if you have something that is known, that is now, at least according to the US patent, they are telling you that it is an innovation that hasn't been done before.
So it's saying
it is something real.
Yeah, they've got something over here, so this is serious stuff. So then other people in the field pay attention, and they may have contributions to.
Exactly.
I see.
And research is collaborative at the end of the day, I alone could not have done it. They alone could not have done it. It is a combination, and to take it to that next step, we would need extra funds, extra resources, extra specializations that we may not have available right here at UNF. And it will allow us to get access to those.
So you could present your findings to someone else or a big corporation or somebody who would have a resource that you could use to help with the study. I see. But so not only do you have this patent on the control for that protein, but you're also looking for a second patent. What is the second one for?
The second one is a little more specific. So these generic Lego bricks that I talked about, right? So the way that we connected those together and which Lego bricks we put together, that's the second patent. It's saying that this particular molecule is the one that we found that works best. So it's really kind of protecting that a little closer, yeah.
Okay.
So that is something unique that you do not find anywhere else in the literature.
Okay, and I want to make sure I also understand something. Are these what you have that is stopping and slowing down and killing cancer cells? Is that something that has come out of a body, or is this a compound that you've created using outside tools, medicines, drugs? I'm not sure the proper term to use.
These chemists have done it. That's their work.
It's not a bodily thing. It's something that's made outside.
Right.
Modeled after.
It's mimicking something that's found in the body, but they've made it.
Okay.
Yeah, they're synthetically made in the lab, and they're meant to look like things that might naturally be found in the body, which is where they find their sort of affinity for the target. So it's kind of like, I liken it to sugar versus artificial sweeteners. Sugar, has certain effects, but it also has the calories, where artificial sweeteners give you the same effect without the calories, same idea. So it looks like something natural, but it doesn't have all the metabolism and that sort of thing. And so it mimics what might otherwise be naturally found in some places.
Okay.
But it's synthetically made in the laboratory.
How much time and effort goes into this? I mean, because I know you all, some of you are teachers and you have other things going on and a life. But how much do you pour into this? And is it something that you pour into after you realize, oh my gosh, we have something, and you dive in? How does that time management work?
I mean, it takes a long time, and I think we'll all say that. So, I mean, I started in 2012, and this was the project that I kind of started with. And about 2020 is about when this kind of took off, right? So it took a lot of years to get to that point. Even in that time, the amount of time that I spend in the lab varies day to day. Some days it may be 1 hour, some days it may be 8 hours. But there's also a number of students that contribute to that, that all take a part in this to help get us to that same point. So it's not that one of us is doing all this. It really is like maybe we're helping lead those projects forward in different labs, but ultimately we're training students to say, okay, here's the next step. Here's what you need to think about. Here's what you get to do. So they get to they spend a lot of time in the lab, and they are absolutely a big part of this.
How many students are we talking about? Maybe at any one time or over the time?
Over the years, it would be several, but there were two main students that were also first and second author on this paper that had done a lot of the work, that had spent hours and sometimes literally 12, 14-hour shifts collecting these cell extracts, creating the compounds, looking at them, working with them at a time. But any given time in any one of our labs, I believe there'll be 2-3 students at least working on a given project, and they'll be spending significant amount of their time there, not only doing active research, with the cells or with the compounds, working with them, but also doing literature review, learning about what is already known about them, and using that knowledge to then critically think about this with us and develop this project further.
So how do the students benefit? I understand that they are getting this knowledge, they're getting this chance to work with you, and boom, they are now going to be, onto something huge.
There are proteins. There are worker bees.
But do they get class credit? Do they get paid? How does that work? Is it volunteer? For students, what does that mean for them?
Some of all of what you suggested, it depends on what their goals are, where some of them just want the experience. Some of them have no intentions of going on to be scientists. They're maybe pre-professional, but The research experience, most people don't understand the process, and I think it's good experience for them to, a lot of them come in very excited and they find out, this isn't for me, or they come in and they find out this is what I want to do and they decide to do graduate school. And so the experience of just doing research, regardless of the specifics, but just learning how the process works, I think is very informative for many of them. But then they get the knowledge. Some of them want to go on and pursue this further in graduate school, and so they get the background of the knowledge and the skill set. Because science, working in the lab, is it's almost like a trade where there's certain skills that you learn that are applicable across any sort of future lab or project that you might work on. So it's really an educational experience.
And that's got to be so fulfilling. Besides the research that you do, to know that you are having an effect on these students who will go on to help solve other things. And I mean, is that part of why you got into teaching or into, am I answering all these questions?
I was at Mayo Clinic before I joined UNF, and one of the things that I missed there, while I had a very fulfilling research life there, was my connection with students, seeing those sparkly eyes full of ideas, and still, they're not jaded yet when they first come in, and they have a different excitement, and they keep you both young and as well as mentally sharp, because they ask you these cool questions that make you think about what we should be doing, what can be done. And that was something I missed. So when I came here, it was a job that I found after the deadline, but I was like, oh, let me just try it out, test the waters. And I ended up getting it, and I jumped on it because it allowed me to go back to, that environment where I saw these fun students that were so excited to be here, to learn, and to be part of these discoveries.
Yeah. And how about you? You chose to come to UNF. What was it? I think I read 2012?
2012, yeah.
Why UNF?
I guess a number of reasons, to be honest. So going through undergraduate, going through graduate school, going through a postdoc, like you experience different things. And when I was in undergraduate, it was medical school. That's where I was going to go. And then I started doing undergraduate research, and I was like, oh my God, I love being in the lab. So I decided to go to graduate school. I went to graduate school, and in that environment, you're allowed to teach some. So like early on, you may teach 1 or 2 courses. In that time, I really enjoyed teaching. But then I kept thinking, okay, I'm going to go work for industry. I'm going to go work for a pharmaceutical company. And I went to this postdoc, kind of another 2-year internship. And in that case, I still absolutely focused on going into industry, and I just missed that teaching. I enjoyed teaching so much, and I was like, I think that's what I want to do. And I knew that I didn't want to go to this top tier schools, and I liked working with the undergraduates. I liked training them and getting them to the point where they get to do stuff like this, right? Undergraduates can be really talented, and I think sometimes people think, well, they're really young. Well, they are, but they also ask these really great questions, and they're really driven by kind of what their passion is. So at that point, I was like, okay, I really want to go somewhere like that, but I want to go to a school that's kind of up and coming. I want to see a university grow. At the same time, so Corey and I go back a long time. So when I was in graduate school at the University of South Carolina, he came and did a 2-year postdoc in the same lab. We overlapped for a bit, for a couple of years, I guess. And then he went off and got a job here at UNF. So a couple of years later, when I was thinking, okay, I'm going to apply for different jobs, he actually reached out. He's like, hey, there's a job in our department you may think about applying for. I jumped on it, and I got the job. And so here we are today.
You got him a job.
I got it to apply.
And you clearly had been here before.
Yeah, so
2012.
2010 is when I started. My experience was much the same. I sort of went through those stages of medical school, not from industry, teaching's where I found the most fulfillment. And so being in a place like UNF that focuses primarily on undergraduate education and teaching classes that are traditionally, students don't like them, right? They're difficult, but they're necessary, like pre-med classes. And so being able to connect with students and sort of help them get through it in such a way that's not so burdensome and onerous. And so that was really my passion, I suppose, is trying to help them not just get through it, but appreciate it for what it was and be successful, but really appreciate the process, not just as a course that must be passed, but to gain some insight and understanding of why it matters, how it matters, and what's the relationship between this class and their ultimate goal, whatever it really is. And so that's always been my real drive is helping students get an appreciation, get through, be successful, and learn material that they're going to use moving forward.
So it's so funny. My daughter, when she was, I don't know, second, maybe first grade, her big thing was science because she hated germs. And like she would walk around like this and I want to touch stuff. I may have had something to do with that. Sure. But she wanted to be a scientist to figure out a way to kill germs. That has passed and she has moved on to other topics. But like, was there something that even got you interested in science to begin with?
So for me, my grandmother had bought this microscope for my aunt when she started med school that she had absolutely no interest in. And so she gave it to me for my 10th birthday. And this was back in Pakistan. And I made slides of anything and everything I could get my hands on. Yeah, like randomly, a thread, a leaf, a flower, a worm, you name it, I did it. And I really enjoyed it. I even got my mom excited about it, who had like nothing to do with this. But she was like, "Ooh, that's fun. What did you find now?" And so I think that's where I already knew that I really enjoyed it. But for the longest time, I also was like, "Oh, I want to go to med school and, treat people with cancer." I still had an interest in cancer even at that time. But then I went to undergraduate and I was in my first job, job was a work study job in a research lab. And then my DIS was in a research lab looking at pesticides and seeing if they were harmful to humans or not, if they caused cancer in humans or not, or so if they were safe to be on the market or not. And I loved it. I could be there all day, all night and not feel tired. I would get out and I'll be exhausted. But as soon as I entered that lab, I was great. Wow. I loved it and I knew it. That was it.
I feel that way about the beach. So, and guys, tell me, what sparked that in you when you were this high?
I don't know that mine, I don't have a definitive story like that. I've always had an interest in understanding how things work, I suppose, but I don't think I ever focused it into, oh, this is my career path, until I was probably in college and taking, the course that I teach now, organic chemistry, and my professor, I'll always remember him, like I just thought it was neat how he seemed to know everything about the chemistry of life. And he wasn't just teaching me the reactions, like, here's this reaction, here's why it matters. Here's where you find it in your day-to-day life. Here, the connections between the world around us and the things he were teaching. And I just always appreciated that. I think that's when I got drawn into this particular field. It's like this guy, like he showed up with a piece of chalk and taught lecture, no notes or anything. I just thought it was amazing how he knew everything and, could relate it. And I think that's where I got sort of drawn into the subject at least.
Which makes sense. I mean, he was able to show how it related to your life. Exactly. So, yeah.
And I think a lot of us, when we find that teacher or, somehow a mentor that is excited about teaching that topic. And for me too, I had my advisor, Greg Orloff at Emory. Oh my goodness. I could listen to him talk about cancer biology all day. Molecular biology all day. He explained it in a way that made it relevant to you, that made it exciting for you to learn, and that was, that gets you hooked.
Yeah, and Brian, did you have?
I probably like a kind of a weird combination of that. So I really do think it comes down to the people, and I think that's probably what draws a lot of us to want to teach. My senior high school chemistry teacher, I remember teaching chemistry, and at the time I was like, I don't get it. I don't understand it. But she talked about how chemistry was this really hard major, and that she only knew a few students who had done it. I was like, okay, fine. Like maybe I could do it. I don't know. My freshman year in college, my very first chemistry class was that professor. I came from a small high school, went to this larger university where my Gen Chem was 200 and so many students, and I walked in. He said, "You should sit in the front row." And I sat in the front row, and I'm overwhelmed, and he kept talking to me. He's like, "You're going to be a chem major. You're going to love it. I can tell you." And it was like just this weird interaction, but like by the time I was like a few weeks in, I was like, I love this stuff. It is great. And so it's just that interaction that just, you're scared of it initially. You're like, I don't know about this. But those interactions very early on just are so long lasting.
In your eyes.
Yeah, I mean, I guess so.
But it's so awesome to see you so excited and passionate about what you're doing. And what you're doing sounds like you were on the brink of getting ready to help millions of people. I mean, that's huge. I mean, is it just me? Do you not see that?
Well, again, cautiously optimistic.
And I'm like, yay, let's do it. But I can understand. Thank you so much for explaining all that. And I hope that inspires someone who may be wanting to study or has diseases and understands how they work. And it's just very inspirational. So thank you all very much.
Well, thank you.
Thank you.
So something interesting happened after that conversation, that discussion with the three researchers. Dr. Fatima Rehman was working to send me pictures and videos of their research projects. And she mentioned that we might not recognize her in the videos because she lost her hair during treatments. Of cancer? You had cancer? So I promptly texted her and I said, do you mind if we talk a little bit more about this personal experience that you've had, especially in light of the research you've been doing? And she said, sure. So during my dinner break, just a second ago, we had that conversation and here it is by Zoom. Okay. Dr. Rehman, like I told you in my text, first of all, wow, congratulations, because I understand you are in remission.
Yes, I am. 1 year this weekend.
Oh, this weekend marks a year? Even
Yes.
Happy anniversary.
Thank you.
Do you mind telling me what kind of cancer it was and
Yes.
where are you now? I know you're in remission, but what are they telling you?
So I had large B-cell lymphoma, which is a type of non-Hodgkin's lymphoma. It is, if in terms of cancer, it's one of the better ones to get than others. I did have bulky disease and I did have to go through chemotherapy for 6 months. And as you know, with traditional chemotherapy, it comes with a lot of side effects. So I am in remission, thank God. I don't have any more cancer that is visible on PET scan, which is great. But I do still suffer from some side effects that are residual from the chemo. And that would probably no longer get any better than they've gotten, including neuropathy and pain, which is, again, exactly why making these targeted drugs is so important for us to make sure that you can give patients not only cure, but better quality of life, both during treatment and afterwards.
And we talked about the different types of cancers and diseases that your treatment that you're working on right now with the other two researchers could help. Is the type of cancer that you had one of the ones that could be affected by this?
Sure it is. It is one of them as well.
Wow.
Yes.
I mean, and if you the irony is not lost on me at all. This is something you've always been interested in, studying cancers. So what was that diagnosis, that news like?
So, you know, I am a firm believer that somehow life gives you a path and kind of guides you on it based on maybe the challenges that you're going to face down the road as well. And for me, because of the knowledge that I had, because of my path and because of my exposure to cancer research, as well as with translational medicine, it made me a more informed patient. So when I did get the news, I had been a lot more scared about it being a much more aggressive cancer. And my initial reaction was actually, thank God, it's not what I feared, but something that is more manageable. And because I had the knowledge, I was able to work with my oncologist, both to understand the therapies better and to get the best options that were out there available to me. So I was able to get into clinical trials that many people would not be aware of right away or would not think to ask about, because there are new therapies coming out every day. There are new clinical trials coming out every day on all types of cancer, for all types of cancer. And it is important for us as patients to also be aware of them, both for our sake and for the sake of humanity as well. even if they don't work on us, they would definitely not be harming us because we'd still be getting traditional therapies as well. But if it does work or if it doesn't, it provides useful information to researchers so they can continue their, research, and they can continue to make better targeted therapies down the road as well.
Now, doctor, I applaud the fact that you are just so open and willing to talk about this. A lot of people keep those kinds of things, close to the vest. It's very private, personal, and different for everyone. But you actually used what your experience was with cancer for your students to learn. Tell me about that.
So, all humans in general, we typically learn based on what is of interest to us and what is somehow relevant to us. And so we were at that time when I was diagnosed, we were just talking about hallmarks of cancer, talking about what makes cancer a cancer cell. We were learning about those specific mutations and everything. And so when I was diagnosed, I used it as an opportunity to bring a little bit of a human aspect to the students. I had told them of my diagnosis that Friday, and I was diagnosed on Friday. So when I came back, I did tell them, this is what had happened. And I said, well, let's look at what type of hallmarks of cancer we can, kind of think about based on this information that I have so far. So in doing so, I was trying to, help them kind of bring a human aspect to it, but think about their information they had learned the week before in a critical manner and use that information, to synthesize a result this time and think about, okay, what type of cancer would it be? Is it aggressive? Is it not? What type of therapy should we think about? So it was just, I used it as a learning opportunity, and in a weird way, kind of used it as it was a way for me to process this information too. So, it kind of helped me bring some balance to it as well.
So you basically used yourself as a guinea pig for the students to kind of
It's all good.
learn on, which is a big deal. Do your fellow researchers, the other two gentlemen, do they know about your cancer?
So they didn't actually know about it right at that time. They do know that I had cancer, obviously, because we do work closely with each other. But at that time, I didn't share this with them. We had just published this paper. We were working on the patent application back and forth and answering those questions. So a lot of time we weren't physically together at that time, and I was still able to continue doing all the stuff that I needed to do with the help of the students that were in the research lab, getting the work done, getting the lab experiments done, and me working with them to analyze it online or in person as I was able. So they didn't actually really find out about it until a little while later, a few months later, when I was in the thicket of chemo and had no hair, no eyebrows, and looked quite, different. But they reached out to me, actually. They heard about it. They reached out to me themselves. And then when we met, it wasn't as big of a shock, but it was good. They were very supportive, obviously.
Well, does your personal experience, how does it change your perspective or change your motivation level? Or, how are you channeling what you experienced
Mhm.
in your
Yeah.
day-to-day?
So, like I said, that I had always been interested in cancer research. I had always been interested in making some kind of a meaningful difference in the lives of cancer patients, either in diagnostic side or in therapy side throughout my career. This gave me a firsthand experience on what they, actually go through. It wasn't just something I read in a book. It wasn't just something that, I heard about through second, third hand accounts. It was something I went through personally. So I can relate a lot better to them. And especially when we are working with actual patients and talking to them about these therapies, clinical trials, I can actually relate back to them a lot more. At a personal level, I can have discussions with them a lot more openly and tell them, yes, I understand what you're going through.
Why cancer? What did you have somebody else in your family that had cancer, or what in the world was the initial interest way back then?
So it was that's a little bit of an interesting but weird story. And so I was in Pakistan. That's where I grew up. And in Pakistan, there was this cricketer who was also our previous Prime Minister, Imran Khan. His mom had passed away from cancer. And he was a bachelor, he was the most eligible bachelor. So everyone was after him, including us, all the 8th, 9th graders in the schools. He was making a hospital, kind of like a St. Jude model, where it would provide free care to cancer patients, both adult and young, who could not afford it because of his experience of watching his mother die in a lot of pain and agony through the cancer process. And so we were all crazy about that idea. I wanted to be, like I said, at one point, I wanted to be a doctor, but I was very much interested in research as well. And when he was going around raising funds for it, I actually met him and I came up to him and I said, I don't have any money for you today, but I promise you, when I am a doctor or when I am, a researcher, I will come back to your institute and I will give you 3 years of my time. And I will work at your place and I will do whatever I can to help you at that time. And I did do that after I completed my PhD. But so that was my motivation. That's what started me on that path. So that's what it was.
That's incredible. I mean, and I wish you continued success on
Thank
your
you.
own health. Yay, remission.
Yes, definitely.
And on your research. I mean, who knows? You may end up sharing your personal story with an organization, a company that can help fund your research.
That would be amazing.
I mean,
Because even if this molecule isn't perfect yet, it is good enough to be moved into the next stage. I know it is able to do very good things. It is able to really affect cancer cells specifically and have an impact on, which course they take. And such molecules combined with traditional therapy allow us to give patients a much better quality of life because we can use lower doses of those traditional drugs that are so cytotoxic, that are so poisonous to the body, while still getting the same effect. And so that would be my hope, that we can at least take this first generation of molecules to the next level and are able to complete, to actual drug in the clinics.
I love your passion, Dr. Rehman. I love
Thank
this
you.
full circle story that you have, and I'm so thankful that you shared it with us.
Thank you. I'm very grateful for the opportunity.